Welcome to the deep dive. If you're a learner preparing for a really intense review, maybe you're trying to cram years of micro and immunology into, a few high impact. Facts efficiency is everything. Today, we are tearing into the absolute essentials from the BRS Microbiology and Immunology 6th edition.
And our mission here is to give you a really clear map of this massive field. We're not just going to recite facts. We're trying to connect microbial structure, genetics, pathogenesis, and the immune response into a framework that you can actually use. This is the stuff that helps with rapid recall when it really matters.
Absolutely. Think of this as your shortcut. This book is designed for retention, right? It uses a bug approach and an organ systems approach.
Right.
We're going to try and blend those, see how the basic science really explains what you see in the clinic.
So let's start where the book does, with the basic classification. When we're looking at microorganisms, we're mostly talking about the protistic kingdom. It's a bit of a catch-all for prokaryotes, eukaryotes, and then the really weird stuff: viruses, viroids, and prions.
But the very first clinical decision you often make comes down to one thing: the bacterial cell envelope. Let's dig into that huge distinction between gram-positive and gram-negative bacteria. It defines everything.
It's the single most important differentiating factor. So take the gram-positive cell. What makes it gram-positive is this incredibly thick peptidoglycan layer. It can be like up to half of the entire cell wall mass.
Wow, half.
A huge amount. And this wall also has teichoic and lipoteichoic acids. The lipoteichoic acids are important structurally. They kind of anchor the wall to the membrane, but both types are also major surface antigens that the immune system sees.
So gram-positive is simpler, but it's tough because of that dense peptidoglycan net. But gram-negative, that's structurally more complex and clinically a lot more dangerous. Why?
It's all because of the outer membrane. Gram-negatives have a really thin slice of peptidoglycan, maybe 2 to 10% of the wall. But outside of that, they have a whole second membrane, a phospholipid bilayer. It's a fantastic permeability barrier. And its star player is lipopolysaccharide, LPS.
And LPS is the part that, strikes fear into the heart of an intensivist.
Exactly. I mean, LPS is the classic endotoxin. When that gram-negative bacterium dies, either from an antibiotic or your immune system, that LPS is released. The toxicity is specifically from the lipid A part. Its release kicks off this huge inflammatory cascade that leads to septic shock.
You also mentioned the paraplasmic space in gram-negatives. It sounds like a tiny detail, but it has huge consequences for antibiotic resistance.
It really does. So the paraplasmic space is that area between the inner and outer membranes. And critically, this is where the cell concentrates its hydrolytic enzymes, especially beta-lactamases, which are just sitting there waiting to break down penicillin before it can even reach its target.
It's an extra layer of defense. Okay, let's shift gears completely to the weirdest agents in the whole system: prions and viroids. Prions seem to just break all the rules.
They're truly unique. They're just infectious proteins, no nucleic acid at all. And yet they cause these devastating CNS diseases like Creutzfeldt-Jakob. A normal cellular protein, PRP, just misfolds, and that misfolded version can cause other normal proteins to misfold. What's really scary is their resistance. They resist heat, disinfectants, nucleuses. You need something like a protease to actually inactivate them.
And viroids are sort of their botanical cousins?
Mostly, yeah. They're mainly plant pathogens, small, circular, single-stranded RNA. But the reason you need to know them is that the hepatitis D virus RNA is considered viroid-like, and HDV needs a co-infection with hepatitis B to even make its envelope.
Which just shows how complex these interactions can get. Okay, let's move on to how bacteria live and adapt. Oxygen tolerance, for example. It's not just a preference.
No, it's about whether they can manage toxic byproducts of using oxygen.
Right, so this is where we get obligate aerobes.
Well,
What lets them thrive in oxygen?
they have defensive enzymes. They have superoxide dismutase, or SOD, and then either catalase or peroxidase. SOD deals with the superoxide free radical, turning it into hydrogen peroxide, which is still toxic. So then catalase or peroxidase immediately breaks that down. Without those enzymes, the bacteria would just be killed by the oxygen they're trying to use.
That's survival. Now let's talk evolution. Bacteria are masters at acquiring new genes, especially for resistance. We have to understand the three big mechanisms, starting with conjugation.
Right. Conjugation is often called bacterial sex, but it's a one-way transfer. It needs physical contact, usually through a sex apellis. This is how they rapidly transfer big chunks of DNA, like F plasmids, or, more worrying for us, R factors that carry drug resistance genes.
The conjugation is the physical pipeline. Then transduction is more like the unwitting delivery driver, right?
Precisely. It uses a bacteriophage, a virus that infects bacteria, as the vector. There's generalized transduction, where the phage accidentally packages random bits of host DNA. And then there's specialized transduction, which is much more targeted. It only transfers bacterial DNA that's right next to where the phage DNA integrated into the host chromosome.
And that's so key. If a toxin gene happens to be right there, it can turn a harmless bacterium into a killer. This brings us right to chemotherapy. The whole principle is selective toxicity.
That's the core idea. The drug has to hurt the microbe, but leave the host, relatively unharmed. And the classic target is the cell wall, which our cells just don't have.
Which is where penicillin and the other beta-lactams come in, hitting that thick gram-positive armor we talked about.
They do that by inhibiting cell wall biosynthesis. They go after the penicillin-binding proteins, the PBPs. These PBPs are enzymes that do the final cross-linking step to make the peptidoglycan wall rigid. Penicillin blocks that. The cell wall becomes weak, and the bacterium esplices.
Okay, so if we move inside the cell, we can target protein synthesis, exploiting the difference between their 70S ribosome and our 80S ribosome. How do tetracyclines do that?
Tetracyclines bind to the smaller 30S ribosomal subunit. They're bacteriostatic, so they stop growth. They work by physically blocking the aminoacyl tRNA from binding to the acceptor site. Basically, they stop the building blocks from coming in, and protein synthesis just grinds to a halt.
So once we have a suspected infection, identification is the first step. The Gram stain is obviously the cornerstone. But some of the most critical organisms, like Mycobacterium, are basically invisible to it.
Correct. That's because of their waxy mycolic acid cell wall. You can use the acid-fast stain, of course, but a high-yield modern alternative is fluorescent microscopy with dyes like auramine rhodamine. They bind to that waxy wall, and they fluoresce so brightly that it's often faster and more sensitive to spot them.
And once you have a culture, you can use these rapid enzyme tests to really narrow the field.
Absolutely. Two major ones. First, the catalase test. It immediately separates Staphylococcus, which is catalase positive, from Streptococcus, which is catalase negative. Second is the oxidase test, which differentiates most gram-negative rods from the huge Enterobacteriaceae family, your E. coli, your Klebsiella, which are all oxidase negative.
So that's the ID. Now let's talk about how they cause disease, specifically exotoxins. These things are incredibly potent.
And many of the most famous ones use the AB subunit structure. Think of it like a guided missile. The B subunit is for binding. It determines which host cell the toxin will attach to. Once it's bound, the A subunit, the active part, gets inside the cell, and that's the part with the enzymatic activity that does the damage.
And a perfect example of this is diphtheria toxin. But its production is conditional, right? A bacterium doesn't just decide to make it.
That's the critical detail. Diphtheria toxin is only made by strains of Corynebacterium diphtheriae that have been infected by a specific bacteriophage that's carrying the tox gene. This AB toxin then goes on to ADP ribosylate elongation factor 2, or EF2, that completely shuts down eukaryotic protein synthesis. Cell death.
For a purely neurological example, we can look at botulism.
Right. Botulinum neurotoxin causes a flaccid paralysis. The toxin gets absorbed, travels to the neuromuscular junction, and it's a protease. It actually cleaves the proteins that are required for acetylcholine vesicles to fuse with the presynaptic membrane. No acetylcholine release, no muscle contraction.
Let's wrap this section up with evasion. How does a bacterium like Neisseria gonorrhoeae stay one step ahead of the immune system?
Neisseria is a master of antigenic variation, specifically with its pili. It keeps a huge library of silent pilin genes in these pilus loci, and it's constantly shuffling gene cassettes from the silent loci into the single active expression site, pili. This creates millions of antigenic variants, so the immune system can never mount a sustained specific attack. That's why you get chronic infections and no permanent immunity.
All right, shifting to viruses, it seems like their patterns of infection can be very different.
We usually categorize them into 4 patterns. There's acute, like measles, where it's cleared quickly. Latent, like herpes simplex virus, hiding out in neurons. Chronic, like hepatitis B, where the virus is always detectable. And slow, like HIV, with a long incubation period.
So when the host fights back, what are the 2 main strategies against a virus?
They have different roles. Humoral immunity, making neutralizing antibodies, is all about preventing the infection in the first place. Blocking attachment, blocking entry, but once the virus is inside your cells, the infection is established. Then you have to rely on cell-mediated immunity. Cytotoxic T lymphocytes, NK cells, they are crucial for clearing out those already infected cells.
And for diagnosis, when culture isn't really an option.
We often look at serology. You're looking for a significant, a 4-fold increase in antibody titer between an acute and a convalescent sample. But for a real-time diagnosis, especially with something like meningitis, nucleic acid amplification tests like PCR are vital for detecting viral DNA or RNA right in the sample.
Moving on to fungi. They're eukaryotic, like us, which makes them tricky to target.
It does, but there are key differences. Fungal membranes have ergosterol, not cholesterol. That's a perfect target. So amphotericin B works by binding to ergosterol and literally punching holes in the membrane. In contrast, the newer echinocandins go after the cell wall. They inhibit glucan synthesis, which is essential for the wall's integrity. Without it, the fungal cell just lyses.
And a quick word on parasitology, specifically the protozoa. What's the difference between trophozoates and cysts?
So the trophozoate is the active feeding motile form. It's delicate, though. It can't survive stomach acid, so it's not infectious if you ingest it. The cyst is the opposite. It's the sturdy, resting infective stage. It has a thick protective wall, which is how it survives the fecal-oral route. It can handle the stomach acid and the environment outside the host.
All right, let's bring it all together with immunology and practice. Let's start with the complement cascade. If you're deficient in a key component, what happens?
Well, losing C3 is a catastrophe. You get broad recurrent bacterial infections because C3 is so central to everything. But a loss of the terminal components, C5, C6, C7, C8, or C9, is linked to a very specific vulnerability: recurrent meningitis caused by Neisseria meningitidis.
Why Neisseria specifically?
Because those components, C5 through C9, are what form the membrane attack complex, the MAC. The MAC is what physically punches holes in the bacterial membrane. And Neisseria, being a gram-negative with a relatively thin wall, is highly susceptible to it. So without the full MAC, your body can't clear that specific pathogen effectively.
Okay, let's look at antibody structure. We talk a lot about IgG, but IgA is the champion of our mucosal surfaces. What lets it survive in the gut?
Secretory IgA is released as a dimer, joined by a J chain, but the key protective part is the secretory piece. This piece is part of the transport receptor that gets IgA across the epithelial barrier, but just as importantly, it wraps around the antibody and protects it from being degraded by all the proteases in your gut and airways.
And for IgG, if you cleave it with papain, what high-yield fragments do you get?
Papain cleavage gives you 3 pieces. 2 identical Fab fragments, that stands for fragment, antigen binding, and then 1 FC fragment, the crystallizable fragment. The FCN is what does all the work: activating complement, crossing the placenta, binding to phagocytes.
To wrap up, let's do a rapid-fire review of the 4 hypersensitivity reactions. Type I is the fastest.
Type I is immediate. It's IgE-mediated. Think anaphylaxis. IgE on mast cells gets cross-linked by an antigen like ragweed pollen, causing rapid degranulation, instant itching, and rhinorrhea.
Type 2 is cytotoxic.
Right. This is antibody directed against antigens on a cell surface. The classic example is hemolytic disease of the newborn, or Goodpasture's syndrome, where antibodies attack basement membranes in the kidney.
Type 3 involves circulating complexes.
Yes, type 3 is from circulating antigen-antibody complexes that get stuck in tissues and cause inflammation. Think of the glomerulonephritis that can follow a strep infection, where these complexes lodge in the kidney.
And finally, type 4 is the delayed one.
Type 4 is different because it's T-cell mediated, not antibody mediated. It takes 24 to 72 hours. This is the mechanism behind contact dermatitis, like the rash you get from poison ivy.
That was an incredibly dense deep dive. We went all the way from the bacterial cell envelope to the complexities of our own immune defenses.
And if you take one thing away, it should be that connection between structure and clinical consequence. Whether it's the toxicity of LPS or how an antibiotic works, understanding the why is the key to mastery. It stops being just memorization.
And to reinforce that, let's leave you with one final review exercise.
A 4-year-old child presents with a petechial rash, high fever, and severe neck stiffness. A spinal tap confirms bacterial meningitis. If labs later show this patient has a congenital deficiency in complement factor C8, which specific organism is the most likely cause? And based on the immunology, why is this patient specifically predisposed to that particular infection?
Ponder that connection, how a single missing piece of the immune system can open the door to one specific devastating pathogen. That knowledge is what turns you from a learner into a practitioner. Keep exploring those connections.